Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com Inside Scientific and Cell Signaling Technologies webinar titled Proteomics to Decipher Biology for the Drug Development Pipeline. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to have Dr. Jeffrey Silva here with us today, who's a principal scientist at Cell Signaling Technologies, who will explore the role of mass spectrometry-based proteomics on drug discovery, biomarker discovery, and gaining a deeper understanding of disease mechanisms. And a little later on, Dr. Matthew Stokes, Director of Proteomics at CST, will join us for the Q&A. And without any further ado, I'm very pleased to welcome Dr. Jeffrey Silva. Jeff, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you very much for the introduction. First, I'd like to thank Scientist.com for hosting this webinar, and for everyone that is online watching. In this talk, I will present how cell signaling technology is enabling rapid and efficient biomarker discovery by simplifying the proteome to quickly focus in on the relevant proteins or pathways that drive a biological response of interest for any given model system. Here, we see why the proteome is so highly complex. Starting with the genome, which contains 20 to 25,000 genes, the transcriptome grows to approximately 100,000 transcripts by considering translation from alternative promoters, splicing variants, and RNA editing events. The complexity of the proteome expands to well over 1 million proteins, including proteoforms, after translating the transcriptome into its corresponding inventory of proteins and allowing for the wide range of post-translational modifications, a portion of which is listed here, that occurs to regulate protein activity, function, localization, and protein-protein interactions. Cell signaling technology has addressed the challenge of the inherent complexity of the proteome by developing an array of different immunoaffinity capture reagents called PTMScan products. These products enable one to simplify the proteome by enriching for specific PTMs or PTM motifs to efficiently focus in on the relevant biology and pertinent biomarkers that are responsible for driving a particular cellular response, whether it be in the context of developmental or disease biology, or due to compound treatment or other perturbations. CST has been developing proteomic tools and providing proteomic services for close to 25 years to address biomarker discovery needs for pharma, biotech, and academia. To provide this type of service, CST is prepared with a well-equipped mass spectrometry laboratory with expert operators, specialized bioinformatics support, extensive translational biology experience with a wide range of sample types, deep knowledge of cellular biology, delivering results within efficient timelines, and complementary products and services for downstream validation or custom assay development. For all our proteomics product users and service customers, your success is our success. We aim to move your research forward. We provide full access to our proteomic expertise. We strive to deliver clear results and specialized technical support. We offer convenience and confidence to our customers, acting as an extension of their lab. And we're focused on providing high-value proteomic solutions. Carrying out a service project with CST is a simple process. We have experience with many types of biological specimens. You simply send us your flash-frozen samples on dry ice, and CST's Proteomics Service Group will do the rest. We start each project with a pre-study consultation to establish and finalize the experimental design. Once we receive the specimens, we carry out the sample preparation as quickly as possible. We provide a complete data package with a guide to help navigate through the results. We deliver quantitative results with recommendations for the best candidate markers, along with supplemental bioinformatics. We carry out a post-study consultation to review the data package and assist with the interpretation of the results. And we expect to maintain a long-term partnership with our clients to ensure they get the most from their study. With nearly 25 years of experience, the Proteomics Service Group has worked with a wide range of sample types, with tools and applications to efficiently identify biomarkers involved in drug treatments, disease progression, developmental biology research, cellular signaling, and targeted protein degradation, including the ability to monitor on-target and off-target effects. Here, we show several types of proteomic service applications that are available for us to survey the proteome in your model system, which includes global proteomics, where we can quantify global protein level changes in a range of sample types across many sample conditions. PTM proteomics, where we can identify and quantify PTM changes between control and test conditions for a wide variety of post-translational modifications. And organelle-based proteomics, where we can profile protein level changes in organelle-enriched samples such as membrane, nuclei, mitochondria, and other cellular compartments. Sample requirements will vary depending on the application and specific goals of the project, and can range from 10 micrograms to 10 milligrams of total protein. The applications we offer allow us to monitor global protein changes for up to approximately 6,000 to 12,000 proteins. In addition, we can carry out PTM profiling experiments where we can monitor modulation of many types of post-translational modifications that can range from roughly 2,000 to 20,000 sites depending on the PTM, the sample type, and treatment conditions. CST can utilize label-free or TMT workflows to perform global protein profiling to identify and quantify protein levels across different conditions. Here we see a generalized workflow for TMT modified from the article highlighted at the top left of the slide that includes lysis, reduction alkylation, digestion, TMT labeling of individual samples, sample pooling, fractionation to obtain deep proteome coverage, LCMS and MS3 analysis, and the corresponding bioinformatics analysis to produce the list of peptides, proteins, and relative quantification values with statistical metrics to assess reliable changes between control and treatment conditions. With this information, the data can be examined using Harkle clustering or Volcano plots to quickly identify those proteins that are differentially regulated between control and treatment conditions, as you can see here in the highlighted areas of the Volcano plot. In this slide, we see the results from a study that was performed at CST, provided on our website from the link at the top left, to illustrate the information you can obtain from a global protein profiling study. The study was designed to perform global protein profiling across 14 cell lines to demonstrate the depth of coverage and the quantitative information that is available from a multiplex TMT experiment. On the left is a heat map of the 14 cell lines with quantitative analysis for approximately 8,000 identified proteins. Below, you can see a zoomed-in region of the heat map with identified proteins listed and the corresponding relative quantitative values across the 14 cell lines. To the right are several examples of follow-up Western blot data from the protein lysates of the 14 cell lines, including FOXA2, CD3, and ERK1/2, along with the relative abundance data from the mass spectrometry-based global proteome analysis for these proteins, showing very nice correlation. Looking at this data, one can appreciate the value of the deep quantitative proteome analysis and how this information from these types of experiments can be very helpful to quickly identify protein biomarkers in any type of life science research environment. Over the years, CST has assembled an extensive toolbox of affinity capture reagents, a portion of which is listed on the left, that we can use to probe a wide range of post-translational modifications in conjunction with the standard bottom-up proteomics workflow to examine important biological processes and cellular signaling. The methodology is referred to as PTMScan and allows one to leverage the specificity of these affinity reagents to simplify the proteome and focus on specific PTMs to discover biomarkers that are modulated within a given system. PTMScan proteomics is used to obtain deep coverage of the proteome through efficient targeting of PTM peptides to produce actionable data of cellular signaling to help understand mechanisms of disease progression or drug resistance or to identify unique biomarkers that will uncover new biology. In this example, taken from published work by CST, a compound treatment study was performed using MKN45 cells, a gastric carcinoma cell line that is known to be MET-driven, which were treated with either SU11274, a selective MET inhibitor, or staurosporin, a non-selective protein kinase inhibitor. The two sets of scatter plots that are shown represent the results from a phosphotyrosine enrichment and a phosphoserine threonine motif enrichment. The x-axis displays the mass of the observed enriched peptides, and the y-axis corresponds to the associated fold change for each identified peptide. The yellow bar represents the threshold region between plus and minus 2.5-fold change, while the green circles represent upregulated phosphopeptides of greater than 2.5-fold change, and the red circles correspond to downregulated phosphopeptides of less than negative 2.5-fold change between compound treatment and control conditions. We can see in the PY results there are a lot of downregulated phosphotyrosine peptides and a much lower degree of downregulated phosphotyrosine peptides in the staurosporin treatment. Although not as dramatic as the PY results, we see the opposite trend with the phosphoserine threonine profiling results, with the majority of the downregulated peptides occurring in the staurosporin treatment. To the right of the scatter plots, you can see the results from an ingenuity pathway analysis for proteins associated in nuclear signaling for both the SU11274 and the staurosporin treatments. The proteins in the pathway are color-coded in relation to their corresponding fold changes, up being green and down being red. For those proteins that had different sites that were differentially regulated, you will see both red and green coloring. Comparing the two pathway figures, it is clear that nuclear signaling is affected differently between the two compound treatments, and therefore these types of experiments can be very useful in establishing differences in off and on-target effects of different drugs, and can also help to lead to a better understanding of the mechanism of action for any particular drug. In addition to developing reagents that allow one to probe different subproteomes, CST has developed a method to probe various cellular compartments. In this example, we see the global proteome analysis from 350 tumor specimens and 80 normal tissue samples, with replicates from each sample type that have been processed through a proprietary membrane enrichment method to identify surface proteins that could serve as druggable targets for cancer therapeutics. The pie chart shows the breakdown for the different cell and tissue types examined in this study. From this work, approximately 2,600 cell surface proteins were identified, and the scatter plot on the right shows the relative abundance of one protein, as shown on the y-axis for EGFR, across the collection of samples listed in the x-axis. Similar studies like this can be performed for other cellular compartments that may be of interest, such as nuclei, mitochondria, exosomes, and other cellular compartments. In my last introductory slide outlining the proteomic efforts and services at CST, I'll describe the work done by CST scientists Clarissa Rakova and coworkers. This is an example of how PTM-directed proteomics discovery experiments can lead to the identification of novel biomarkers for clinical diagnostics that can directly influence patient treatment and outcome. In this work, a global survey of tyrosine kinase activity in non-small cell lung cancer was performed to identify novel disease drivers. Phosphotyrosine profiling was performed to enrich a total of 4,551 phosphotyrosine peptides to approximately 2,700 proteins from 41 cell lines and 150 tumors. The analysis identified the tyrosine kinase ALK within the top 10 candidates due to its elevated levels. Further investigation revealed a fusion of the N-terminus of EML4 with the C-terminus of ALK in some cell lines and tumors. Additional work showed that 3 to 7% of non-small cell lung cancer patients express the EML4 ALK fusion protein in their tumors, suggesting it to be highly oncogenic. Authors showed that cancer cells expressing the EML4-ALK fusion protein, are sensitive to the small molecule ALK inhibitor crizotinib, and in 2011, the FDA approved crizotinib for the treatment of ALK-positive non-small cell lung cancer patients. As follow-up to this work, CST developed a highly specific and sensitive rabbit monoclonal antibody which detects the EML4 ALK fusion. In collaboration with Pfizer and Ventana, the FDA approved an IHC diagnostic test using the ALK antibody from CST to help select patients for crizotinib treatment. For the next series of slides, I will highlight some work in a few papers that have used PTMScan products as tools for their proteomic studies. In the first article, a large effort with Bernhard Kuster, co-workers and collaborators was conducted to integrate total and PTM proteomics in the context of dose and time-dependent experiments as a workflow to help understand the mechanism of action for a number of different classes of drugs. I will highlight a few examples in this work, but will encourage everyone to read the article as it is a wonderful demonstration of how proteomics can help accelerate drug discovery and how it could be extended to serve as a powerful tool in advancing developmental and disease biology. The workflow described in this article, called Decrypting, is comprised of the following steps. Cells were treated with increasing concentrations of drug for a fixed period of time, or a time course experiment was performed with a fixed concentration of drug. Cellular protein was extracted from the drug-treated cells. Extracted proteins were digested with trypsin. Peptides were labeled with TMT for each drug dose or time point, and then pooled for subsequent LC-MS analysis. PTM peptides were enriched by immunoprecipitation for acetylation and ubiquitination, or mobilized metal affinity chromatography was used to enrich for phosphopeptides. Multiplexed LC-MS analysis was performed to produce peptide protein IDs and TMT-based quantification. Custom scripts were used for fitting dose response curves, filtering significant drug perturbations, and creating a variety of visualizations. To the right, we see the number of total peptides and proteins identified within the entire study. With regard to the protein summary, there were a total of 15,713 proteins identified for total proteome analysis, 11,331 phosphoproteins, 3,006 ubiquitinated proteins, and 1,377 acetylated proteins. On the left of this slide, we see a schematic representation of the different sample sets outlined in the manuscript, which was comprised of 31 drugs from 6 different drug classes and 14 cellular model systems. I will be highlighting data from only 2 of these studies, the biologic study, which consisted of 3 drugs, and the protein turnover study, which included 2 drugs. On the right, we see a schematic representation of the type of data that was collected in this study, which consisted of 1.8 million drug dose response curves obtained for proteins or post-translationally modified peptides by LC-MS. For phosphorylation, ubiquitination, represented by GG or gly-gly, or acetylation. For each peptide, PTM site, or protein, the half maximal effective concentration, or EC50, and the corresponding cellular effect size was determined from each of their corresponding dose response curves. In traditional single-dose experiments, significantly modulated peptides are identified by comparing replicate experiments of drug treated versus control in a t-test. A Volcano plot is a common way to visualize the data and identify significant changes in peptides by relating the biological effect size, indicated on the x-axis as the log2 fold change, to the statistical significance of the effect noted on the y-axis as the minus log p-value. The Volcano plots in this figure show the results of the phosphoproteomic experiments using different concentrations of the kinase inhibitor, afatinib, in the EGFR-driven carcinoma cell model A431 with 4 replicates. Regulated phosphopeptides are highlighted, red is down, blue is up, in the case where they exceed a fold change of 2 times and a p-value of 0.05 threshold, as indicated by the dotted lines. As one would expect, the number of drug-related phosphopeptides increases with drug dose. However, at the relevant dose of afatinib in this cellular model system, at the EC50 value of 10 nanomolar, merely 3 phosphopeptides survived rigorous statistical testing. This is in contrast to the many phosphopeptides that are actually regulated by afatinib at this concentration and illustrates the difficulty of selecting a meaningful drug concentration for a volcano plot analysis of drug-perturbed phosphoproteomes. At the top left, we see a volcano plot analysis for the kinase inhibitors dasatinib, afatinib, and gefitinib at a common concentration of 10 micromolar. Downregulated phosphopeptides are shown in red and upregulated phosphopeptides in blue throughout all panes of the figure. Just below these panels, we see the Decrypting potency plots of the same drugs in which the PEC50 values for each phosphopeptide, as derived from the corresponding dose-response curves, are plotted against the effect size observed, or fold change, at the maximum drug concentration used of 10 micromolar. These plots not only directly relate drug effect size and potency, they also highlight if a drug affects one or several targets with similar or different potency. This is illustrated by the single distribution of the PEC50 values for the selective EGFR inhibitor gefitinib, while we can see 2 distributions of the PEC50 values of the multikinase inhibitor dasatinib. The bottom scatter plot shows the same PEC50 values with respect to the minus log of the corresponding fold change. To the right, you see an overlay plot of the in-cell potency distributions of phosphorylated sites regulated by the same drugs in A431 cells to illustrate the drug cell profile presented in this work. Here, the rightmost panel shows the same in-cell potency EC50 data, or drug cell profile, as shown with the overlay plot to the left, but now grouping the regulated phosphopeptides by kinase substrate sites of annotated signaling pathways within the Phosphocyte Plus database to generate what is referred to as the drug pathway profile in A431 cells. In this illustration, we can see that the majority of the potency of afatinib is driven by modulation of EGFR, ERK, and AKT signaling, with some effects caused by the modulation of the p38 MAPK signaling pathway. This is in contrast to what we see for dasatinib, which has the majority of its potency driven by modulation of SRC and ephrin family signaling, with some lower potency modulation within the EGFR, ERK, AKT, and p38 MAPK signaling pathways. In this slide, I'm showing you the results from the analysis of 2 covalent proteasome inhibitors. In the left panel, we see a bar plot showing the number of dose-dependent modulated phosphopeptides at a given time point for both bortezomib and carfilzomib treatment of RPMI 8226 cells. The Venn diagrams at the top and bottom of the bar plots illustrate the number by size and overlap of phosphopeptides between the 2 drugs at each given time point. Here we can see that the number of regulated phosphopeptides increases steadily throughout the 16-hour time course. The circled Venn diagrams below illustrate the total shown by the dashed line and the regulated fraction shown by the colored circles of phosphopeptides and phosphoproteins detected for the 2 drug treatments. In the right panel, you see the same type of results, but for the corresponding global protein analysis. One interesting point to make here is that there are many more phosphopeptides and phosphoproteins that are differentially regulated than with the global protein analysis. As we saw in the previous slide, the number of regulated phosphorylation sites drastically increased over time. Based on those differentially regulated phosphopeptides, in phosphoproteins, there was a very strong activation of cellular stress. Among these changes was the potent and strong upregulated phosphorylation of the serine 248 site of ATF4, the master transcription factor of the integrated stress response. The ATF4 serine 248 site showed an EC50 value of approximately 100 nanomolar and was modulated by about 20-fold. In addition, the serine 98 site of the small heat shock protein HSPB1, which is both an activator of the proteasome and an inhibitor of apoptosis, was also dramatically affected upon both drug treatments. For the purposes of this presentation, I've only reviewed a few examples from this work. However, I would highly recommend reading through the other examples to learn how the overall strategy of integrating total and PTM proteomics can be used as a useful tool in guiding drug discovery efforts. I've also included more details about the drugs that were evaluated in each case study and the cell lines that were used in this recent manuscript. In this article, Dr. Albert Heck and coworkers, in collaboration with CST, demonstrate how an efficient serine-threonine motif antibody enrichment strategy is used to interrogate cyclic AMP-dependent kinase signaling in Jurkat T cells upon stimulation with prostaglandin E2, as opposed to the more general immobilized metal affinity chromatography approach. On the left, we see how the phospho-PKA substrate motif antibody, which is designed to recognize the PKA substrate sequence motif RRX phosphoserine phosphothreonine, was used to screen control and various time points for PGE2 treatment. The Western blot was performed with Jurkat cells stimulated with PGE2 at 10 micromolar over a 60-minute time course using the phospho-PKA substrate antibody shown at the top and a tubulin antibody shown at the bottom as control. Below, we see the densitometry analysis of the Western blot. From these results, the 1-minute and 60-minute time points were selected as test conditions along with the DMSO-treated control. To the right, we see the general schematic for the study showing the sample preparation, starting from the control and treated cells to the lyse digested peptides, to the dimethyl labeling of the lyse peptides from each sample time point, to the PKA substrate phosphopeptide enrichment, and subsequent LCMS analysis. On this slide, we can see the comparison of the phospho-PKA substrate motif results compared to a previously published immobilized metal affinity chromatography method, or IMAC, for serine-threonine phosphopeptide enrichment with the same cells and conditions by Maya and coworkers. On the left, we see the Venn diagrams representing the overlap between the phosphoproteins and phosphopeptides identified between the two methods. A total of 2,908 phosphoproteins with the IMAC and 238 phosphoproteins from the phospho-PKA substrate motif enrichment, with 146 phosphoproteins in common between the two methods. At the peptide level, we see a total of 10,558 phosphopeptides with the IMAC and 288 phosphopeptides from the phospho-PKA substrate motif enrichment, with 22 phosphopeptides in common between the two methods. Given the little overlap between the two methods, the targeted method is very specific, enabling access to low-level PKA substrate phosphorylation sites. The phospho-PKA substrate motif is recognized by the AGC family of serine-threonine kinases highlighted in the bottom right branch of the human kinome map shown on the right, with the substrate motif of RRX phosphoserine phosphothreonine as shown in the slide. Although the IMAC enrichment was able to enrich for more phosphoserine-threonine peptides, the phospho-PKA substrate motif antibody was able to target those specific kinase substrate motifs within that specific branch of the human kinome to help discover new kinase substrates. A total of 306 high-confidence pKa substrate motif-containing phosphopeptides were first grouped into 5 different clusters based on their time-dependent relative abundance profiles, 3 with an upregulated pattern, 1 group with a PGE2 insensitive sites, and 1 grouping with a down-regulated profile. The color coding highlights the signal intensity windows of the representative peptides for each of the 5 clusters, with blue as the control, red as the 1-minute PGE2 treatment or intermediate label, and green as the 60-minute PGE2 treatment or heavy label. On this slide, we see a pKa-focused temporal view of the protein-protein interaction map derived from both the string database and the substrate lines were derived from the search function in the Phosphocyte Plus database, with the color coding indicating sensitivity to PGE2 stimulation. As expected, several of the identified phosphoproteins are involved in the canonical pathway of pKa as directed by the pKa substrate motif antibody. Interestingly, the network analysis reveals the presence of many other proteins not previously linked to cyclic AMP prostaglandin E2 signaling, yet containing a reliable pKa consensus motif that is phosphorylated upon PGE2 stimulation. These proteins are therefore likely substrates and belong to a range of different protein classes and functions, illustrating the widespread role of pKa in other biological processes. In the last example, Rivera and Udeshi at the Broad Institute, in collaboration with CST, describe a workflow using the magnetic bead format of the Ubiquitin remnant motif PTMScan product, referred to as HS for high sensitivity, coupled with the Kingfisher robot to configure an automated sample processing workflow for Ubiquitin PTM profiling studies. Here we see a schematic of the manual and automated workflows from start to finish, including enrichment, to on-antibody TMT labeling and washing, to enriched KEGG peptide elution and stage tip cleanup prior to LCMS analysis. Summarized here, we see that the manual workflow for processing a TMT10 experiment with agarose beads takes about 27 hours, while the manual workflow with magnetic beads takes 3 hours. By utilizing the Kingfisher Flex instrument with the magnetic beads, the sample processing for a TMT10 experiment is reduced to 2 hours. Not only did the sample processing time be reduced by approximately 14-fold, but the throughput was increased by 6-fold, from 16 to 96 samples per day for a TMT sample preparation. To benchmark the performance of the automated UbiFast workflow, parallel sample processing was performed for 4 TMT10 experiments with A375 cells. The reproducibility and performance metrics are summarized to the right of the workflow schematic. The stacked bar plot shows the percentage of fully, partially, and unlabeled KEGG peptides, with an overall TMT labeling efficiency of close to 90%. The side-by-side bar plot shows a consistent number of KEGG peptides identified and quantified from each of the TMT plex channels of approximately 17,000, and about 2,000, or 12%, non-modified peptides accounted from nonspecific binding. The violin plots show the distribution of CVs between differentially labeled TMT channels for each experiment, with an average CV of approximately 20%. Overall, the automated workflow showed an increase in the number of identified and quantified ubiquitinated sites, improved reproducibility, and significantly reduced processing time. The automated method significantly reduced variability across processing replicates compared with that of the manual method. To summarize, I'd like to point out that CST offers a wide range of proteomic services to address many different applications, from quantifying protein abundance among different samples, to global PTM profiling to monitor changes in a variety of different post-translational modifications that may be perturbed in your experimental system. In addition, CST offers PTMScan kits to carry out targeted enrichment of critical PTMs and enable customers to perform these types of experiments with the mass spectrometry instrumentation they may have in their own laboratories or available to them through their proteomics core facility. Lastly, CST has created a new class of PTMScan reagents that include a magnetic bead format that is compatible to automated workflows, using robots like the Kingfisher system to enable high-throughput processing of PTM-centric proteomic screening studies. For those people who do not have a Kingfisher system in their laboratory, we have established protocols for integrating PTMScan workflows into the assay map Bravo for both peptide enrichment and C18 cleanup. This concludes my presentation. I'd like to thank everyone for joining in on this webinar to learn about CST's proteomic services and products, and I look forward to taking your questions during the Q&A session.
Fantastic. Well, thanks so much, Jeff, for a great presentation. And with that, I'd like to invite Dr. Jeffrey Silva and Dr. Matthew Stokes back on the line for the Q&A session. All right, then. Ready to answer a few questions, Jeff and Matthew?
Yeah, absolutely.
Looking forward to it.
Awesome. Yeah, me too. So we'll kick things off with a nice question here. What is the advantage of using an antibody enrichment approach for biomarker discovery instead of an unbiased total proteome workflow?
I'm happy to take this, and please, Matt, add to this if you think I've missed anything. I think this was, something that I tried to cover in the presentation. The idea being that the antibodies are designed to hone in on that PTM site or PTM of interest, and it allows you to enrich for those, cast a wide net and enrich for those particular sites or proteins that are modulated with that PTM to link that to the disease or the mechanism of action for the drug. I'll leave it at that, but I think, Matt, if you have anything else to add, go ahead.
Yeah, sure. No, that was really good. I mean, I guess the first thing I would say is, we like to do both, right? So we do a lot of total proteome work. We think that's a valuable component to any kind of, discovery experiment. But yeah, like you were saying, Jeff, I really think, when you're talking about mechanism of action, or you're talking about, signaling pathways that may be activated or perturbed by a particular treatment, right? A lot of times you really need to be looking at the PTM landscape to really get a sense of what's happening in that biological system. So I really like it that way. And then it's a case of really depth of coverage too, right? So, just a total proteome experiment is not going to tell you anything about phosphotyrosine. And so, doing an antibody-based enrichment and getting real depth of coverage of, for example, the tyrosine phosphoproteome is something that can be really important for some facets of biology.
Fantastic. Yeah, great answer. All right, next one here. What type of proteomic service offering would you recommend for someone interested in prioritizing a series of drug candidates for targeted protein degradation?
Matt, I'll let you
okay.
start off with this one.
Sure, I'll take this one. Yeah, so TPD is really hot right now. A lot of people working in the area of targeted protein degradation. We're really excited about it as well, and some of the opportunities that the technology is bringing to the fore. What we're seeing a lot of people doing for targeted protein degradation space, number one is total proteome analysis, obviously, right? Looking at protein level changes and being able to quantify changes of thousands of proteins in your samples. But another one that people are really excited about doing in the TPD space is ubiquitin proteomics, right? For obvious reasons. And Jeff showed some great examples of people leveraging our ubiquitin enrichment technology. So that's a great option as well when you're looking targeted protein degradation, seeing what proteins are being differentially ubiquitinated and even the sites of ubiquitination in response to treatment. And then the last thing that we also do is IPMS type experiments, right, where you're pulling down a particular protein, via an antibody, or maybe you have a compound with a handle on it that you can pull down and see what proteins are binding to that compound, binding to that protein of interest in your samples, and how does that binding change across different treatments.
I think the only thing that I would add to that is, with CSTs, PTM antibodies, they are compatible for Western blots. So for people that don't have an idea of what type of PTMs are sort of engaged in the mechanism or in the disease progression, you can survey a number of these PTM antibodies using under Western blot conditions to find where you see modulation of a particular PTM, whether it be a serine threonine motif, ubiquitin modulation, acetyl-lysine, phosphotyrosine, and then that way you can quickly hone in on those PTMs where you see maximum changes and follow up with a proteomic experiment to discover those new sites or those sites that are modulated.
Fantastic. All right, another good question here. Can your proteomics service use FFPE clinical samples? And do you also provide service for surface or membrane protein extraction followed by MS for clinical samples?
Yeah, so I guess I'll jump on this one. So the short answer is yes. So we've worked out some really nice methods for doing total proteome analysis on FFPE samples. And we've actually done some comparisons, taking the same material, splitting it, doing half fresh frozen, the other half FFPE. And we've shown that you get just about as much data, probably 95 to 98% of the proteins identified from the FFPE samples that you got from the fresh frozen. So you're getting a high quality data set from those FFPE samples, which is really great. We're starting to look at, is it possible to do PTM work from those FFPE samples? So be on the lookout for new and exciting directions there in FFPE. And then for the membrane, yeah, so Jeff had a slide or two earlier in his talk where we do have some proprietary methods for membrane enrichment and then total proteome analysis. Really powerful that it gives you about twice as many proteins with a cell surface or membrane ontology compared to a regular total proteome analysis. So you're able to get better depth of coverage of those membrane proteins, see what's on the cell surface, and make quantitative comparisons across your samples as well.
I think the only thing I'll add to that is that comment about be on the lookout. We'll make an effort to get something posted soon to give people a sense of how many proteins we're able to identify from those FFPE type samples. I'd look for that within a month or so.
Awesome. Yeah, very cool. Glad to hear that you still get such good quality data from it. All right, next question here. So somebody said they're interested in multiple PTMs to help understand drivers for a particular disease model system. Would this mean that I would need to increase the amount of sample to cover the different PTMs, phosphotyrosine, phosphoserine, threonine, and acetylation?
I'll go ahead and start with this, but I think Matt will also have some comments on here. But depending on the PTMs of interest, we are able to perform sequential enrichments, and that's which means that you do not necessarily have to grow or scale up more sample. We can take the flow through from say a PY enrichment and pass that through beads for like acetyl-lysine, for example, or ubiquitin. So that allows you to, for samples that are primary cells or cells that are difficult to work with, you don't necessarily have to scale that up to triple or double the amount for a typical analysis. I'll let Matt go ahead and add his comments as well to that.
Yeah, absolutely. So doing those sequential enrichments, that's something that we do all the time. The only kind of restriction that we put on that internally is we say you really want to be looking at different PTMs when you're doing those sequential enrichments. So, you can set up a whole workflow, phosphotyrosine, acetylysine, ubiquitin, methylation of arginine, right? And do all of those sequentially on the same material. The only other thing that I'd add is on the sample requirements. So our new version of PTMScan, our new version of this method where we cut to peptides and enrich and then run mass spec, this PTMScan HS we call it, we've actually designed that to allow for lower sample amounts. And in fact, most of those we've validated at 1 milligram of input protein rather than 10 or 15 or 20 that we were using in the old days. So that's another big advantage and kind of opens up this analysis to a lot more sample types, being able to use less material for each enrichment.
Good question.
Yeah, great. All right. Does the PTMScan technology make it possible to increase the factor impact of a publication?
I guess I could take a stab at this one. I would say that, given that you're able to utilize the antibody, the PTM antibody, to hone in on the specific biology, it will help you uncover those novel sites that may be unique to your model system, and therefore, with those results, could justify that work being published in a high impact journal. Without that enrichment, it would be more difficult to reveal that from an MS experiment, and so that's my immediate response. I'm sure there are other, but Matt can add to that.
Yeah, I mean, I guess we can't guarantee anything, right? But but yeah, I mean, I think, like Jeff said, it's the ability to get those unique answers from doing these more specific enrichments that you might not get from just a regular total proteome or just an IMAC enrichment for phospho, and then also, getting to, I would say, actionable data faster, right? So doing that, you're doing something that perturbs tyrosine signaling, being able to get a really in-depth look of what's going on, what changes are happening to thousands of, tyrosine phosphorylated sites is really powerful and really will, I think, kick up the impact of the research that you're doing and help get you to answers faster.
I'll add one more thing to that, and I refer the person who asked the question to look at the article, the serine 3, the SU11274 staurosporin article that I referenced in the talk. If you look in that publication, you'll see also a plot of the protein changes that happened in that drug treatment with the MKN45 cells. And what you'll see in that plot between control and treatment for both drugs is there are not many proteins that change. So if you were doing a protein-centric method, you would not reveal much biology there if you just looked in that direction. Yet, having done the other PTMs, we uncovered a number of proteins and modification sites that were regulated by those drug treatments. And so that's just sort of an example of if you're doing an experiment and there's a rich amount of data that is relevant to the biology, that will help you build up that story for that high impact paper.
Yeah, great answer. All right, so what experimental strategy would you recommend to compare cell secretomes, especially in treatment conditions that result in differences in cell number, for example, comparing monoculture and co-culture?
Matt, I'll let you take a stab at this one.
Yeah, sure. So, we've done some secretome type experiments before, and I think, it's you can get really interesting data from it. I think that what the question is kind of leading to or asking about is, what's the proper way to try and normalize across samples when you're doing these kind of experiments? Another great example of this is you may have treatments that make big changes to cell size, right? And so, protein content. So normally we normalize and just have the same amount of total protein for each condition that we go into these experiments, right? And for 90% of the studies that we do, that is an appropriate way to normalize across samples. There are other ways to normalize across samples. So if you do a cell count before the samples are sent or before the samples are processed, we can normalize by cell count. And we can actually look at these things even after the data has been processed. You can apply different normalization strategies and look and see where is your data appropriately centered. We also include controls when we're doing these type of analyses. We came out with this whole set of control peptides. These are really cool. That it's 3 peptides for each PTM, and you spike them in before you do your enrichment. And now, after you do the enrichment, when you run the mass spec, you can see the signal from those 3 peptides. They're heavy isotope labeled, so you can distinguish them from the endogenous peptides. And you can really get a nice measure for how consistent that enrichment was across all the samples. So that's another great tool for making sure that everything was nice and consistent across your samples, you've normalized in a proper way, and that you're getting, again, reliable, actionable data out of the study.
Excellent. All right, we were talking about sequential enrichment maybe a question or two ago, but does sequential enrichment have any disadvantages to it?
I'll take a stab at this one. I would say that from a if the question I think is sort of looking into whether or not you're sacrificing the quantitation, we can tell you that we've done many of these types of studies, and as long as you choose the right PTMs and they don't overlap, it will not have any detrimental effect on the quantitation. In terms of other disadvantage, maybe, you are limited in terms of what you're able to do for sequential. So for instance, you might not want to do similar serine threonine motifs together because you may deplete out in that first pass the motif in that second pass. So that's where you sort of need to plan for the experiment. And for that particular example where you want to probe a related motif, you may need to scale up and run that as an independent enrichment.
Yeah, I would agree with that. I think, we always try and set up those sequential enrichments to look at different PTMs. I mean, there was an example, this is going way, way back now, but we did sequential enrichments with 2 serine threonine motif antibodies that had different motifs, right? One had, for example, an arginine at -3 and then phosphoserine threonine. The other one had proline at +1. But what we saw was that some of the peptides were actually depleted from that second enrichment because they had an arginine at -3 and a proline at +1. And so that's why we always feel safer to say, okay, you can do a phospho, then you can do an acetyl, then you can do ubiquitin and methyl. As long as you're looking different PTMs, there really is no disadvantage.
Yeah, and the other thing I would add here, and this has been done, it was actually done in that paper with the SU11274 and staurosporin. If there are related motifs, you can actually formulate the enrichment to include those serine threonine motifs you want to capture. So rather than running them as separate IPs, you could make a cocktail of those motifs that you want to focus on and do that as a single enrichment.
Absolutely.
Excellent. Yeah, great answer. All right, I think in the interest of time, we'll just make this next question the last one. But what advantage is there to utilize PTMScan methodologies for biomarker discovery as opposed to global protein profiling or IMAC methods?
Yeah, sure. So, I think we talked a little bit about, the total proteome, and Jeff gave an example from that paper of, not much is, you're doing this early time point, not much is changing on the protein level. IMAC is another thing that we like and we do a lot. It's really a great way to get a broad survey with a single enrichment of serine threonine phosphorylation. But we've seen plenty of examples, and the most obvious of these is tyrosine, where you just simply don't get good depth of coverage of low abundance proteins or low stoichiometry sites with that IMAC analysis, right? It's great for a broad survey. When you need the depth of coverage, like with tyrosine, you really need to be doing that more specific enrichment. So I think that's the real power of these. But I'll tell you, a lot of people will do both or will do all three, right? So we do plenty of projects where people will do phosphotyrosine to get the depth of that signaling space. They'll do an IMAC enrichment to get breadth and get, you know, more global survey of serine threonine phosphorylation without going too deep. And then a total proteome analysis to see what's happening to protein levels in the background.
All right, fantastic. Well, Matthew and Jeff, thanks so much for joining us today and for your insights. It's really been a pleasure having you with us.
Great. One last thing. In that, for those people that were watching, I forgot to mention that our email addresses were provided. And if you do have any questions, please feel free to send us out an email, and we'll get back to you as soon as possible.
Yeah, definitely. If you do have any questions, definitely you can feel free to send them to the email addresses there. And in closing, we hope you enjoyed the Scientist.com, Inside Scientific, and Cell Signaling Technology webinars, and we'll see you again next time. Have a great day, everyone.
Thank you, everybody.
Thank you.